So, okay, good evening. We'll start in 1 or 2 minutes. So, right, it's Labor Day, but as you know, we are laborers that we don't get a day for ourselves because our day is getting closer. So, we have to keep on working. So, let's have a nice paper discussion on Labor Day. And yep, so paper class is going ahead as with the usual program. We have a one structured essay to discuss from paper number 6. So we'll discuss that. So today's plan is to discuss that structured essay and then discuss a few MCQs from paper number 7. So I got a bit delayed to upload the paper number 7, but it's not the plan to discuss that paper entirely today, right? So you are given time to answer paper number 7 until next Monday. Okay, you are given time to answer paper number 7 until next Monday, and online people, you can upload it to the website. And if you come to ISM on Sunday, you are able to answer the paper, right? From early morning to till evening around 7 p.m. you are able to answer. But our revision class 26 is from 1 to 5. So if you are joining the revision class, you can join. You can write the paper afterwards or before. Okay, so you online people also, you are given time to answer the paper until Monday. I will give time till Tuesday maybe. Okay, and your paper 6 papers will be marked and uploaded very soon to the drive, back into the drive, so you are able to download them. Okay, so because of these holidays there was a slight delay in marking these papers because sort of the paper marking, the panel requested some holidays because they just finished A levels, right? So they were a bit out of reach in the month of April. So I couldn't blame them because they should be enjoying their result as well. So it's that time is going to come for you very soon. Okay, so next year you can celebrate everything, but this year it's the year to work. Okay, right? Okay, so let's quickly go through the this structure is a number 3. All of you, can you hear me? All good, right? So we have a bit of genetics question. Okay, so we have a bit of genetics from this because I am now including everything we have covered in the revision class in my papers. So it's easier to mention what I am not including. Okay, what I am not including in the in the paper class is microbiology, environmental, and applied. Right, that's all I'm not including because we have covered molecular because we started the revision class plan with the sort of grade 13 focus. Okay, so we have covered in great detail molecular, genetics, and grade 13 nervous, endocrine, reproduction, and support and movement we have covered. There's going to be a month long program for microbiology that is going to be covered using question based. Okay, right? So that is so microbiology will be covered very fast. Okay, so those are the content that I'm going to include. So from this question is from genetics. So what are they asking? Okay, right, let's get into the question. Define the following terms: recessive allele, epigenetics, dihybrid cross. Okay, what do you mean by a recessive allele? So, you know, in the very first page of the sort of the starting page of your genetics, you have a set of definitions. We wrote down those definitions. When we were doing genetics, we went ahead with like a small note with the series of definitions and doing some example, right? So, what is the definition of a recessive allele? What is the definition of a recessive allele? Yeah, you can say there's two things I want to write down there, right? A recessive allele is, I mean, You know, a recessive allele, you can define it in two ways, but it's the only one way is given in your resource. A recessive allele is showing its face apitamu then the allele that is determining the allele that is determining the tall stem, right? So there are possibilities, no? You can have capital T and simple T alleles. So capital T is the dominant allele, T is the simple T is the recessive allele. So simple T produces short stem, right? If you want to have the effect of short stem, you need to have the recessive allele in homozygous state, right? If you have this form of genotype, that is the heterozygous state, it is going to be completely masked. There is no effect. So that's the definition that we are writing here. The allele which does not exhibit any noticeable effect on the phenotype at heterozygous state. The allele which does not exhibit any noticeable effect. No noticeable effect at what stage? At heterozygous. There is a effect if the allele, if the recessive allele is homozygous, but in heterozygous state it is going to be masked, right? Without a noticeable effect in heterozygous state. That is a recessive allele. Then what is the definition of epigenetics, right? So epigenetics is like karame, right? When you can't explain something by a certain set of principles, by a certain science, you say, okay, it is outside the syllabus. It's like that. Whenever you can't explain a certain phenomenon by the current sort of genetics principles, we say, okay, those things are out of the sort of the scope of genetical study, right? Things that are not controlled strictly by what? The direct genetic principles, we call it outside the genetics or epigenetics, right? So you can say occurrence of certain, so it's a study, right? Occurrence of certain phenotypes of certain characters controlled by factors other than their DNA or genetic code. Okay, occurrence of certain phenotypes, okay? Occurrence of certain phenotypes of certain characters controlled by factors other than DNA or genetic code. So you have to, I'm going to give marks, okay, like this. Certain phenotypes controlled by factors other than DNA or genetic code, I'm going to give 2 marks. And recessive allele, may make a definition here, I'm going to give 2 marks there as well, a noticeable effect in heterozygous state. Then, what is a dihybrid cross? Right? What is a dihybrid cross? You are crossing two hybrids. For example, this is a dihybrid. I'm sorry, this is a monohybrid. What is a dihybrid? Right? For example, with the tall stem, if you are covering the studying the shape of the seed by capital R and simple R, capital T simple T, capital R simple R. This is a dihybrid. It's hybrid for two features. Hybrid in relation to two genes. That is dihybrid. So, what is the definition of a dihybrid? Yeah, what is the definition of a dihybrid? I need to give you the exact. Yeah, dihybrid cross. They are asking. Okay, they are asking a dihybrid cross. Breeding experiment conducted between two organisms with heterozygous condition for two specific characters is dihybrid. So, it's a breeding experiment. So, you have to say it's a breeding experiment, right? Two organisms having heterozygous, very important, heterozygous for two characters. For two characters is a dihybrid. Okay, for two characters is a dihybrid. Clear? Okay, then all right, plant. Okay, next question. Plant and animal breeding cause significant, okay, major impact on world agriculture. State two such major impacts. Okay, impacts of animal and plant breeding. So these are sort of the coming under the applications of genetics, right? So see, they can be questioned in certain situations. So what are they asking? Animal breeding on agriculture. Okay, okay. Agroeconomy. Okay, impacts. Right. So you can write down. There's one, two, three, four. Addressing world feed and food quality needs, addressing food supply needs for a growing world population, adapting to environmental stresses, satisfying industrial and other end-use requirements, developing animal and plant varieties with aesthetic values. Right? Those are the um, set of, let's say, impacts that you can have, sort of the effects you can produce by the breeding. Name two agricultural crops produced from seeds which are habitually self-fertilized. Right, habitually fertilized. There's a list: self-fertilized: wheat, oats, barley, and tobacco. Self-fertilized means what? Within the same plant, you take the pollen and deposit on the same ovule. Right, you deposit on the same ovule. So that comes under what? Self-fertilization. Habitually self-fertilized. That is inbreeding, right? So yeah, yeah, coming under the concept of inbreeding. Okay, coming under the concept of inbreeding. So any two, any of these, wheat, tobacco, barley, aromeo, right? So it is coming under inbreeding. Okay, self-fertilization. So it can be within the same plant. So within the same plant, of course, always within the same plant. It can be from the same flower pollen into the same flower ovule, or it can be another flower on the same plant pollen onto another flower ovule, but always within the same plant. So same plant means that is within the same organism, right? Self-fertilization or inbreeding always applies to within the same organism, right? So I recently did a seminar before the 2028 batch started for this 2028 body small button, right? So you know, in O level syllabus there's a small error with this self-fertilization. So in A levels we learned what is proper self-fertilization and what is proper cross-fertilization, right? So these are usually habitually self-fertilized, right? What is meant by mutation breeding? What is meant by mutation breeding? So you know, under the genetic principles, right? Principles of that are sort of genetic principles used in breeding. You have one, two, yeah, three, right? You have three genetic principles, right? One is producing a polyploidic state. What do you mean by polyploidic? That means you have more than two, 3n, 4n, 5n, 6n. Again, you have several ploidy copies. That is polyploidy. What is the second principle that you can do? That is this mutation breeding you can cause. Okay, you can cause mutation breeding. Final one, that is genetic modification. Genetic modification means kelima, it is molecular techniques. You are going to take out the DNA, you are going to do some modification outside the cells, then you are going to insert them back. Okay, so but here they are asking what is mutation breeding, the definition. Okay, so right, so what is the definition that we can take? The method of inducing desirable mutation in crop plants using either chemical or physical agents. Okay, sorry. Inducing. Okay, it is always inducing. What the word you cannot miss is desirable. Right, you cannot miss the word desirable mutations in crop plants. Right, the mutations, of course. What we are trying to do is have a positive output, no? Right? So whatever the mutation you are trying to cause should be a desirable mutation in crop plants using, so mutagens, right? The agents causing mutation are mutagens, can be physical mutagens or chemical, Can you tell me some examples for physical mutagens? Yeah. So the chemical agent is given. Chemical agent is given. You can take sodium azide, sodium azide, ethyl methanosulfate, right? You can, any one of those, you are going to get 1 mark, right? So if you haven't studied this, I recommend you take out the resource book. Now itself you highlight these things. Okay, this is the first time you are, this is, take it as one of the times that you are going to study. So study with me, right? There's going to be, I mean, there is going to, you are having less and less time to study these again, right? So always think of it, this is the last effective time that you are going to study these. So what are the chemical agents used to induce mutation? These two you can take. Okay. Okay, two physiological features which have been developed by mutation breeding in crops and ornamental plants. So what have we, what have we managed to cause by mutation breeding? So mutation breeding is a very, very difficult procedure, because proper mutations, let's say these uh, proper mutations, proper means desirable mutations are very, very rare. Desirable mutations are very, very rare. Therefore, out of, I think there's a, yeah, 0.01 is the frequency of having a proper mutation out of, sorry, it's like 1000, 1 out of 1000, right? 1 in 1000. Okay, so you can induce mutations, but most of the time they are deleterious or even silent. Okay, so beneficial mutations are very, very rare, but we have managed with constant experimentation and also sort of crossbreeding with the proper ones, we have managed to cause, some positive outputs. What are they? um, Flower color, seed size, crop yield, disease resistance and salinity tolerance, drought tolerance and early maturity. Okay, so those are the things that we have managed to uh, sort of positively change. Okay, any two of those: flower color, seed size, crop yield, disease resistance. Okay, and yeah, so your paper, yeah, paper 6 has been uploaded. Okay, if you upload your written papers, you can now download. Okay, what is the why is the genetic code considered as a triplet code? Bit of a tricky question. Okay, why is it considered as a triplet code? What are the answers that you have written for this question? This is so we are jumping to a bit of molecular now. So we are going to a bit of molecular. These questions are easy, no? These are just asking sort of theory. Theory means sort of things you can just memorize, right? From the applications of genetics. Now we are going to a bit of molecular, chuttak, sort of theory plus application questions are being asked. So why is the genetic code a triplet code? Yeah. What is the answer? What is the genetic code or triplet code? You can formulate an answer to this. Okay, you can formulate an answer to this. So the answer is the language of nucleic acid has 4 letters, the language of protein has 20 letters. Okay, a combination of nucleotides is needed to code for one amino acid. Okay, therefore amino acids are coded by triplets of nucleotides, hence genetic code is a triplet code. Can you understand? I repeat the answer again. The language of nucleic acid has 4 letters. What are they? A, T, G, C. The language of protein has 20 letters. Those are what? The 20 amino acids. Right? So if one of these nucleic acids only coded for one amino acid, then you are only going to have 4 amino acids coded, right? So with the nature, with evolution, we have decided we are going to have 4 into 10 to the power 3, okay? 4 into 10 to the power 3, that is due to having a triplet. That means 3 letter combinations of these 4 letters. 3 letter words of these 4 possible letters, you are going to end up with 64 words. These 64 words are going to be called as codons. These 4 letter words are called codons, right? So in your molecular biology, page number, molecular biology, page number 17, this translation chart is given. This one, yeah, how these 3 letter triplet words codes code for the 20 amino acids. This chart you don't need to remember, but by studying that, you know, there are 64 codons, which is much higher than the 20 amino acids, which is much higher than the 20 amino acids. Therefore, what has happened? More than one codon code for a per amino acid. That means a certain amino acid is coded by more than one codon. Up to about 4 maximum codons you can have, right? Up to about 4 codons you can have per amino acid. Okay. Then there are 4 special codons. 4 special codons, 3 stop codons are there. That is going to indicate, okay, stop making a protein when you have these 3 stop codons, and there's one start codon, that is AUG. Okay, AUG. AUG is the start codon by protein synthesis starts in August. Protein synthesis starts in August. Okay. State 2, okay. So did you understand? Okay, I'll repeat again. I'll write the answer. So the language of nucleic acid has 4 letters. Language of protein has 20 letters. Therefore, a combination of nucleotides is needed. This is the word I am looking for. A combination of nucleotides is needed to code for one amino acid. Okay, I'll send you the marking scheme as well. A combination of nucleic acids. So 1, 2, 3 points. Okay. Understood? Next question, state 2 differences between transcription and DNA replication. Transcription and DNA replication. What is transcription? What is transcription? That is DNA driven RNA synthesis. You are looking at DNA and making an RNA. What is the mRNA? Specifically an mRNA. DNA replication, what you are doing? By looking at DNA, you are making DNA. You know? By looking at a DNA, you are making another DNA. So don't overcomplicate this. Okay, transcription is something in protein synthesis, DNA replication is something entirely different. Don't worry about it too much. Write down the things you know. Write down the things you know. Okay? First thing you can write down, you can write down in, you can say in transcription, okay? In transcription, the product is a RNA. The product is a RNA. In replication, the product is a DNA. So those things you can write down. Then what else you can write down? Okay, now a bit something specific you need to write down. In transcription, you need to write down only one strand. One strand is the template, because why? You are looking at one. So DNA has two sides, no, two strands. You are taking out one strand and using that as the blueprint or the template. So in transcription, one strand is used as template. Okay, in transcription, one strand is used as template. And in replication, what is happening in DNA replication? You are going to make another copy of the DNA that you have right now. Neither. So, in replication, what's going to happen? Both strands act as templates. Both strands act as templates. Both strands act as templates. Hari? What are my differences? So per one comparison, I'm going to give 1 mark each. Okay. Any doubts you have? Any doubts? Clear. So some of my molecular recordings are available freely in my YouTube channel, and the others are available as study pack, right? But okay, so you can access the freely available ones in the YouTube channel as well. Okay. Any questions up until now? My advice is, when you look at a molecular question, do not get, do not panic, right? Do not panic. So right now is the time to, first thing, if you are avoiding keeping, I mean, if you keep avoiding a certain topic, the first thing to do is to sort of master that section. You need to stop being nervous or anxious about that topic. Okay, first think, okay, I have enough time. If you say you don't know molecular, you don't know micro, okay, there's still more 3 months left. There is more than enough time to cover that section, right? So the first thing is to remove the anxiety or the panic about a certain section. Then you slowly, from one end of the lesson, from the beginning of the lesson, you slowly start to study it, right? You slowly start to study it, because this is not the time yet to panic. දැම්ම එහෙම ලොකුවට panic වෙන්න ඕනේ නැහැ. You can still work in an organized manner. Sort of that steady and organized is going to win this race, not the panicked cramming. Okay, the panic cramming is not going to be productive, right? You can do that panic cramming the night before the exam, not right now with 3 months before the exam. It's not going to be productive, as well as it's unhealthy and not very useless. Okay, right. What is a recombinant vector? What is a recombinant vector? Now vector is what is a vector? So in DNA recombination, listen, okay, in DNA recombination, what are you going to do? You are going to take DNA from one cell, from an organism. You are going to take another DNA from another organism. You are going to take a useful part of one organism. Okay, imagine it's an insulin producing gene or a disease resistant gene, whatever. you are going to take that useful gene from one organism and you are going to put it into another organism, so you that the second organism is going to have that feature. Okay, that's what you are going to do. But the problem in recombination is what? DNA of living animals or cells of living animals, they don't like to take foreign DNA. වෙන තැනකින් ආපු DNA ගන්න කැමති නැහැ. They don't like foreign DNA because they know it's evolutionary defense, right? So in recombinant DNA, you have to sort of, what do you have to do? You have to sort of fool the receiving cells into accepting foreign DNA. You have to fool them. So for that, we use vehicles that are called vectors. We use vehicles that are called vectors, right? So a vector is a vehicle carrying foreign DNA. So that vehicle, when you have combined it with our sort of modified DNA that we want to put it into a cell, it's a recombinant vector. So the definition is a vector carrying foreign DNA is a recombinant vector. A vector carrying recombinant DNA. A vector carrying recombinant DNA is a recombinant vector. Alright. So they have given some examples for recombinant vectors. They have given some examples. So we study some examples for vectors in your syllabus. We study about plasmid vectors. What are plasmids? If you take a bacteria, right, other than their main DNA that is found in the nucleoid region, other than their main DNA that is found in the nucleoid region. Then plasmids කියන එක මතකයි නේද? Right? These are basic things. Other than their main DNA found in the nucleoid region, certain, I mean, bacteria, they have sort of other independently replicating DNA sequences, that are called plasmids. They are sort of unrelated to the main nucleoid DNA. So this is a typical bacterial cell, no? A typical bacterial cell. So all this is your main nucleoid DNA. I'm going to select it. So, right, this is your main nucleoid DNA. What we use as a vector are these. These blue color में देखाते. These are your plasmid DNA. You can use them as vectors because they are independent of this main nucleoid DNA. They are independent. So you can use those as vehicles, vectors. Okay? That's what we are trying to do. You got it? So you can have several types of, okay, I'll explain what is this yeast artificial chromosome. So yeast is not a prokaryote, the yeast is not a bacterium, right? Yeast is not a bacterium, but yeast also have these plasmid-like DNA pieces in there. Yeast is a fungus, you know. Yeast is a fungus. So yeast can have these plasmid-like pieces of DNA as well. But because the yeast is a fungus, which is a eukaryote, listen carefully, it's a eukaryote, we don't call them as exactly plasmids, we call them yeast artificial chromosomes. Yeast artificial chromosomes because they behave like bit like chromosomes even though they are sort of plasmid-like. They have several benefits. One thing, as I keep on telling, yeasts are eukaryotes. So yeast artificial chromosomes are in the eukaryotic system. So that is a benefit because if you want to use that yeast artificial chromosome as a vehicle to send some DNA into another eukaryotic cell, so the yeast being a eukaryote is a benefit. Okay, eukaryotic system එකේ. So it's good. Then if you think of it like this, Apple computers right? MacBooks it's easier to transfer information, right? Other than an Android and an Apple device. Right? Because they are both the same operating system. Not Android, Windows or Apple, MacBook or Android or Apple. Eukaryotic system is much easier to sort of manipulate or exchange things. How does it? Anyway, so one thing is yeast artificial chromosomes are big. The other thing, yeah, big. Hinda, imagine the yeast artificial chromosome is like a bigger lorry. So because it's a bigger lorry, you can fit more sort of recombinant DNA to be transported. One thing, they are operating in the eukaryotic system. The other thing is they are large. Okay. Right. Come back to the paper. Yeast artificial chromosomes, they are large. So a large amount of, okay, I'm sorry, right? They are large, therefore a large amount of DNA can be copied. Or you can write any, or you can write, can be copied, you can write they operate in the eukaryotic system. They operate in the eukaryotic system. Then bacteriophages. Bacteriophage, can anyone tell me what is a bacteriophage? The chat is very inactive. Can anyone tell me what is a bacteriophage? What is a bacteriophage? What is a bacteriophage? Is it a bacteria? Yes. A bacteriophage is a virus infecting bacteria. A virus capable of infecting a bacteria is a bacteriophage. Okay. So they operate like this. A bacteriophage has so many benefits, so many uses that we have discovered with molecular technology, right? We have used bacteriophages for so many techniques, right? I'll show you. Yeah, exactly. I can show you the exact technique of how bacteriophages are used to transform. What do you mean by transformation? A transformation is when the receiving cell takes up the DNA that we are trying to give. So this is this. This is a bacteriophage, um, sort of mediated gene transfer, right? So what we are going to do, this is the donor cell. I told you, in any DNA recombination technique, there should be a donor that we are taking the DNA from. So here what we are going to do is, we are going to need this allele. These two alleles we are going to take. The A positive and B positive alleles that we are going to take. That is from the donor cell. Okay. Then the virus, what's it going to do is, when it infects, this entire donor cell's DNA will be broken down. Then what's going to happen is, virus is going to replicate inside this donor cell, and certain virus particles are going to take the genes that we want. They are going to take the genes that we want. Then when you infect a recipient cell with that virus, what's going to happen? This virus with the gene that we want is going to inject that into the recipient cell. So now what you can you see? This recombinant cell has the gene that we wanted in its DNA molecule. Okay, that is how a virus can be used to transmit a gene or a piece of DNA from one cell to a donor cell to a recipient cell. Got it? Find it, I mean, when you know what is happening, you can memorize it. Ekapaattama bacteriophages කියනට වඩා, right? Any questions? Any questions? Okay, right. Okay, come back to the paper. Bacteriophages, so what do they do? Bacteriophages, what do they do? You can say vector can be inserted in the host cell by mechanism of infection of the bacteriophage. Right? The vector can be inserted into the host cell by the mechanism of infection of the bacteriophage. This is called transduction. Okay? By the mechanism of infection. A virus always try to infect. Right? Virus tries to infect and copy itself. State two significances of using marker genes in cloning vector. Okay. Right? Then make it that calmly, don't overthink it. Okay. Don't try to, okay, what is a cloning vector? What is a marker gene? Right? So, yeah, think independently. Okay, what is a marker gene? A marker is something that we use to highlight something. Something used to highlight whenever to sort of pinpoint something out, right? In a large number of genes, from a pool of genes, you use marker genes to isolate something, identify someone. Okay. So are we? Why do we need to use a marker gene? Right? It's like this. Imagine you do this. Okay, first thing you need to know, these molecular biological techniques are like karma. They are very easy to be unsuccessful. Okay, they are very easy to be unsuccessful. Their success rate is very low. So what can you do? නිකන් basic logic එකෙන් කියන්න පුළුවන්, if something's success rate is very low, what can you do to improve the success rate, improve the probability of something happening? What can you do? You can do it many times. You can do it in a much larger scale. So these molecular biological techniques are not done in maybe one or two cells. You try to do it into thousands of cells. Okay, you do it for thousands of cells, and among those thousands of cells, at least one or two may be transformed. That is what we are trying to do. So how do you identify, okay, there's a massive colony with thousands of cells? How do you identify? Then imagine there's a petri dish, right? There's a petri dish with so many bacterial cells that you try to transform. මෙම petri dish එකක් තියෙනවා. This is a bacterial colony. How do you identify who is transformed? That is how, that is by, in the transform cells, if you can include a certain marker or identifying factor, then you can clearly isolate them. So you can say you can use marker genes for screening to filter out. So you can say okay, I need to formulate an answer. Okay, significances. Okay, I need to give you significances of using marker genes. Okay, hold on. It's not very nice in my marking scheme. Hold on. Marker genes. Yes. Two significances. One thing, the few colonies, okay, write down if you have written, the few colonies originating from the transformed cells can be screened among many untransformed cells. Okay, the few colonies, again, among this entire sort of the culture, you can extract out the only the transformed cells. So you can say the few colonies originating the few colonies originating from transformed cells, transformed cells can be screened among many untransformed cells. Among many untransformed cells. Then there's another significance. Okay, it's a bit complicated. I know your molecular biology lesson can go into a bit of detail sometimes that are unnecessary. But the thing is, okay, transformation is the cell that you are trying to give this foreign DNA, it has accepted this DNA, but it has not been integrated. So it has accepted, but it has not been sort of නිකන් එයා එයාගේ කරගෙන නෑ එයාගේ අර නිකන් සිංහලෙන් කිව්වොත්, so it has not been properly integrated into its genome. So transformation happens, but the insert is not there. Give me 1 minute, okay. Okay, right. So the other significance is transformation does not necessarily mean that the insert is there. Okay. So what's going to happen is, so you have to say, you need to have another marker to find out if the colony with the vector and so මෙහෙමනේ, so vectors ලගෙත් problems තියෙන පුළුවන්. A vector is like a vehicle. So imagine, so you are trying to send a delivery to about several homes. So මෙහෙමනේ, so imagine I'm trying to deliver my tubes into my students, to my students. Okay, so I am using a sort of a courier service. So those are the vectors, right? Vectors can be bacteriophages or yeast artificial chromosomes, plasmids or whatever. So you are the recipient cells. So my tute is the DNA of interest, right? So the vectors, okay, so I should deliver you the tute by a vector, yes. But sometimes this tute delivery company might just go into your home without my tute. Then the delivery sort of bike එක යනවා without my tute. So the vectors are going into the cell, but they don't have the DNA of interest. So the insert is not there. Okay. So that's why they say Another marker is required to distinguish colonies with the vector containing the insert from those with the vector only. කියන්නේ දැන් so some students might have a sort of a delivery guy coming to their home without a tute. Okay, so that's the problem. So the other thing is to distinguish colonies, with the vector containing the insert from those with vector only. Those are the significances. Got it? Of using marker genes. So marker genes can be antibiotic resistance genes. Okay. So how does an antibiotic resistant gene marker work? Again, when you drop an antibiotic onto this, the cells that do not have this antibiotic resistance gene are going to die. So the others are going to remain because they are resistant to that antibiotic. Okay, shall we continue? What are small tandem repeats? Then, again, what is the trick to studying this? What is the trick to studying this? Molecular. Whenever you have a difficult term, mulimma, at least you need to know where do you have a certain concept? Yeah, it's a technique in, yes, can anyone tell me where do you have small tandem repeats, in what technology, what part of molecular, where do you have STRs? Yes, 27 pep class was much more active. මොකද ඔයගලන්ට වෙලා තියෙන්නේ, right? Yes, DNA fingerprinting. Okay, so in DNA fingerprinting we use a technique or the presence of certain pieces of DNA that are called small tandem repeats. So you can say මෙහෙම කියන්න පුළුවන්, non-coding sequences which are 2 to 6 base pairs, I mean non-coding sequences where 2 to 6 base pairs are repeated tandemly one after the other 100 to 1000 times. Okay, write down non-coding sequences where 2 to 6 base pairs are repeated tandemly, 2 to 6 base pairs are repeated tandemly 100 to 1000 times. If you have written you can say they are determined by PCR using specific primers, that also you can take, but this is the best answer. Okay, what are STR? They are non-coding sequences where 2 to 6 base pairs are repeated tandemly. Okay. State a technique where small tandem repeats are used. That is, of course, DNA fingerprinting. Easy. State two advantages of using STR. Okay, they occur frequently in genome, easily amplified by PCR, highly variable polymorphisms. What do you mean by polymorphism? Means they can have variable, I mean, so many different patterns can be there. So many different patterns can be there. So that's what we mean by highly variable polymorphisms, and large number of characterized STRs are available. So there's four. Out of those four, any two you can write. Occur frequently in the genome, easily amplified by PCR, highly variable polymorphisms, and a large number of characterized STRs are available. Those things you need to write down. Okay. Any questions? Then we have a bit of something to talk about, agarose gel electrophoresis. So I thought of I'll quickly remind you what is being done in this technique, agarose gel electrophoresis. Okay, you don't need to know much about these things, but as I mean, it's sometimes even it's undergraduate level in university. Okay, so even us as medical undergraduates, we didn't even get to perform these things because they are sort of sometimes even postgraduate level techniques. This agarose gel electrophoresis and gene gun, there's not even a, I think gene gun is very rarely used and very expensive procedure, and viral transduction, these are very very advanced techniques even though they have been included in your syllabus, right? Okay, hold on. Electrophoresis, I had a nice diagram somewhere. Ah, yes, okay. I think I gave this in my tute as well, but so this is how this whole agarose gel technique is used. I mean, it is done. This is a very simplified version of this. Okay, gel electrophoresis. So you use a matrix, that is a gel matrix composed of agarose, right? So agarose is sort of a jelly-like matrix. What you are going to do is you are going to cut DNA by DNA cutting enzymes that are called what, sort of restrictive endonucleases. Okay, so you are going to have some pieces of DNA, then you are going to load them into these holes. Those are called wells. DNA is negatively charged. Okay, so what's going to happen is when you apply a positive sort of an electrode on the opposite side, these negatively charged DNA molecules are going to move through this gel. Because the gel is having very microscopic pores, their speed is reduced. So they are sort of moving through these pores. Okay. So DNA molecules to move towards the positive terminal, and after a fixed amount of time. So there's different times of electrophoresis for different purposes. So after some time, then we can add a fluorescent dye to it, and then if you shine some this sort of a UV light to it, you can see the different DNA pieces. So this is the DNA wells. මේ side එක තමයි the DNA wells. So the DNA has moved. So according to their size, they have separated, right? So usually the lighter DNA, smaller DNA pieces are going to travel further, and larger DNA pieces are going to be slower. Understood? No? Simple logic. Okay. So this is gel electrophoresis. After gel electrophoresis, there's another technique, that is southern blotting. I don't have an exact diagram to show you southern blotting. Southern blotting is done so that at the end of electrophoresis, you have this DNA in the gel. No? You have some pieces of DNA in the gel. So what you need to do is you need to take these pieces of DNA out of the gel. So that is actually done using a sort of a weight. You actually squeeze the DNA pieces out into a nitrocellulose membrane. That is called southern blotting. Okay, so you put some weight onto this gel matrix, and the DNA pieces are going to be absorbed into a nitrocellulose membrane, that is southern blotting. Then out of that membrane, you can take the DNA pieces out. You can extract the pieces of DNA. Okay, so what is the main purpose of following techniques in gene technology? You can write down separation of DNA molecules according to size. Very important, separation of DNA according to size. Size කියන එක ඕනෙමයි. According to size. Then B, PCR. PCR is our main way of photocopying DNA in vitro. What do you mean by in vitro? If any DNA technique is being done outside cells, we call it in vitro. That means in a lab condition. Okay? Outside of a living system, in a sort of a outside living cells, you call it as in vitro technique. So PCR is like a big photocopy machine for DNA. You can make large number of DNA copies using a PCR. Okay, so copy sequence of DNA in vitro mimicking DNA replication. So copy DNA, copy DNA in vitro mimicking replication. Mimicking replication, that is the proper definition. Okay, mimicking replication. Then finally, chelating agents used in DNA isolation. Why do you need chelating agents? Because what do you mean by DNA isolation is you are going to break cells and you are going to extract the DNA. You are going to break the cells and extract the DNA. Okay, so if you when you are, so how is the normally DNA, where is the DNA usually? In the nucleoid region of a prokaryote or inside the nucleus in a eukaryotic cell. So there the DNA is protected, but whenever you are breaking cells to take the DNA out, the DNA is going to be exposed. One of the problems is there are enzymes that are nuclease enzymes that are breaking down DNA. So these nucleases require some metal ions, right? Metal ions are needed for this nuclease activity. You know, when you need some other things for enzyme activity, what do you call this? When you need something for the activity of an enzyme. When you need non-proteinous components for the activity of an enzyme. Quickly tell me. When you need non-proteinous components for the activity of an enzyme, what do you call that? Basic metabolism. What do you call that? Non-proteinous components needed for enzyme activity. Okay. My 2027, no, even 20, no, 28 can't. 27 students can say this. Again. Cofactors, right? Cofactors. So these nucleases need cofactors such as metal ions, right? So when you remove the metal ions using chelating agents, what do you mean by chelating agents? They are going to sort of make salts with these metal ions. So the metal ions are going to be removed. So the nucleus activity is inhibited. Okay. So you can write down to remove metal ions needed for nucleus activity. Yes. Okay, there's a question to separate small DNA strands from large DNA strands. Um, uh, I mean, they are small and large. That sounds like we are separating them into two, small and large. It's not like that. Based on their different lengths, you can have different lines. Different lines you can have. So there can be several classes of sizes. So you is exactly not that. We are separating them according to size is the best answer. Okay. Okay, so with that we come to the end of that paper. Then the essay marking schemes will be sent to you. Okay. This nerve impulse transmission. Sorry, this is a chemical synapse. We wrote this in the revision class. Adrenal gland we just covered last week. So adrenal gland I didn't write. No, we wrote a pituitary gland essay in the class. Meiosis we have written, right? So I'll send you the marking scheme. Chromosome mutations. Okay, if you want, I can discuss next week. Photorespiration, very commonly questioned, but I keep on giving it to you because you cannot lose marks in easy essays. Easy essay spelling, you cannot lose marks. Xylem ascent and phloem translocation, stomatal transpiration, and xylem ascentis. Those are the things question. So there's a question to explain the PCR process. To be honest, you don't need to know the process of PCR. Okay, you do not need to know the process of PCR. You can see it is mentioned in the resource, right? You can see it is mentioned in the resource. You don't need to know the process of PCR. Okay, you only need to know uh, the applications of PCR. Okay, you only need to know the applications of PCR. There has been once a question, I think 2020, 2020 or 2021, applications of PCR were asked. Okay. Only things you need to know, what is the primer, the DNA polymerase used, they can be questioned. It is from TAQ DNA polymerase, but we know that already from the DNA polymerase enzymes from the bacteria Thermus aquaticus, TAQ DNA polymerase. ඔය වගේ small MCQ points can be asked, but the process of PCR you don't need to know. Okay. So that is the end of paper 6. Any questions? Okay, so since we have time, let's discuss some MCQs from paper 7, few MCQs, then we can finish for the day. Next week, we are going to have the proper discussion of paper 7. Okay. So I had to explain All right, so I decided to include mainly questions from animal form in paper 7 and some genetics and molecular, right? So it's going to be mainly animal form. You can see it's going to contain everything from up to neuro and reproduction, support and movement, everything, right? Genetics is there, molecular is there, because the best way to remove your sort of fear of molecular is to do questions from molecular. Then again we have some animal form, okay, right? Mostly it's animal form. Then out of the structure, there's four structure essay questions. They are going to be from mainly animal form as well, okay, mostly animal form questions. Some genetics there, bit of pedigree chart and bit of molecular. The paper is long, okay, and the essays are going to be all animal form essays, okay. So you need to, because you know up to 45 marks of the paper, if you write both essays, up to 50 marks of the paper is from animal form. So without knowing, I mean, this is the time to master animal form without overly thinking about other things as well, right? So paper 7 is animal form dominant. So if you have friends also looking for a proper paper class, you can ask them to join, right? Why is it not zooming? Okay, right. Shall we discuss some questions from paper 7? So you can upload your papers to be marked, right? Mainly, I mean, as you need to take the benefit of taking the papers marked, particularly for your SCQs, okay? So if you haven't downloaded the paper, you can download it right now. Okay, right. Correct statement regarding the regulation of digestion in man. Okay. Digestion is in the stomach is slowed down. Okay, slow down. Okay, due to low levels of cholecystokinin and secretin. Cholecystokinin and secretin hormones are secreted in response to what? Secreted, they are secreted in response to what are the main signals? I mean, so okay, if you want to slow down the digestion, that means wooding stomach is in the above, right? Cholecystokinin secretin is secreted by your sort of the small intestine, duodenum. So what is the duodenum saying? I am currently busy, so don't send me food right now, ne? That's what it's saying. So if it is slow down, that is due to high levels of cholecystokinin and secretin secreted by the small intestine saying, okay, I am busy right now. Yes, usually high levels of fat. Fats are a bit difficult to broken down. It can be due to high levels of protein, but you remember it as high levels of fat. Okay. Fatty acids or amino acids in the chyme. Yes, a lot of proteins are difficult to broken down. There is no nervous regulation in the stomach. What do you think? False. Because when the stomach wall is stretched, what's going to happen? You release gastrin hormone. When the stomach wall is stretched, gastrin hormone is released. So gastrin is going to circulate through blood and stimulate the stomach to secrete more gastric juice. Chyme is rich in fat, gastric juice secretion is inhibited. Yes, when the chyme is rich in fat, again you are going to slow down the digestion in the stomach for a longer time. That's why when you eat a fatty meal, you are going to feel sleepy and you feel full for a longer time. Ne? But after the New Year's meal, kiri pani hima kala, right? Biryani fried rice hima kala, right? What's going to happen? You feel full for a long time, right? So that is correct. Churning starts before the stomach, food reaches the stomach. What do you think? Okay, wait. Chyme is rich in fat. Okay. Okay. Churning starts before food reaches the stomach. That is correct. Oh no, wait. Churning starts as the food reaches the stomach. I'm sorry. Yes. Before anyway, I didn't see this, right? As the food arrives, okay? As the food arrives, the churning starts. Cholecystokinin triggers the release of pancreatic juice. Cholecystokinin triggers the release of pancreatic juice. Yes, that is also true. Ne? Yeah, bile from the liver or the gallbladder, and digestive juices from pancreas. Hmm. Yeah, so you can take both answers, no issue. 3 and 5. Both answers 3 and 5 you can take. Okay, shall we move to question number 2? Blood group or groups that can be received by a person with blood group B during a blood transfusion. What is the logic? Whatever the blood that you are giving to a person, he should know that blood. He should know that blood. Okay? That means a blood person. Okay, so this is the recipient. So this is the recipient. He had an accident or something, so he is lying on the bed, hospital bed. So you are going to connect a blood bag. Okay, you are going to connect a blood bag. So he is B positive. He is B positive. So if you take his red blood cell, there's the B antigen. No? If you take his red blood cell, there's the B antigens on his red blood cell. So he knows the B antigen. Eh? And what else does he know? He knows the rhesus factor. Eh? He knows rhesus factor. So I'll draw the rhesus factor in blue color. He also knows the rhesus factor. Okay. So you can give him any blood with the rhesus factor because he already knows the rhesus factor. Wait, hold on. It says only blood group B, ne? Yeah. So we don't know. Okay, sorry, I just assumed. We can't say he knows rhesus factor. So let's assume he only knows antigen B. Sorry, right? He only knows antigen B. Okay, so he only knows antigen B. Okay, blood group B. He only knows the antigen B. Okay, so what can you give him? So you don't know if he is rhesus positive or negative or not. All right. So what can you give him? So you can't take a risk, ne? So he can be B negative. If he is B negative, what is the problem? He doesn't know the rhesus factor. So if you give him positive blood, he is going to have a reaction to that positive blood. Okay. So he is going to have a reaction to that positive blood. So you cannot take a chance. So any option with a positive blood group, you cannot take a risk. Anything with a positive blood group, you cannot take a risk because you don't know the rhesus factor. So what can you give? You can give him B negative, O negative. O negative is O negative. O negative is the universal donor blood. So you can give O negative to anyone. B negative has the B antigen and no rhesus factor. So you can safely give it. Okay. Yeah, correct. Okay. Right. Question number 3, bit of respiration. Okay. Question number 3, bit of respiration. Correct statement regarding human respiration. During inhalation, the gap between the two pleural membranes increase. What do you mean by the pleura? What do you mean by the pleura? What are the two types of pleura you have? What are the two types of pleura that you have? You have sticking to your lung itself, the outer covering of the lung itself, you have the visceral pleura. Then on the outer layer of the chest, so the inner lining of the chest wall itself, you have what? The parietal pleura. So the lungs are located in between those two. Right? Lungs are located in between those two. Sorry, lungs are covered by those two. Visceral pleura covering the lungs, chest wall inner side, you have the parietal pleura. So those two pleura are sticking together by a fluid called the pleural fluid. Okay, so what should happen is, how are you going to make your lungs bigger? By making the chest wall bigger, chest cavity. So with that, when the chest wall expands, because these two pleura are sticking together, the lungs are going to be expanded as well. During the respiration, inhalation, the gap between pleural membrane doesn't increase. They are going to stick together. If the gap increases, the lungs are going to stay the same size. During internal respiration, what do you mean by internal respiration? The respiration happening in your tissues. That means blood flow is going like this. Then there are some tissues right here. These are your body tissues. What's going to happen? From the blood, oxygen is unloaded. From the tissue, carbon dioxide is loaded. Make any path. Unloading of oxygen and loading of carbon dioxide occurs. Total lung capacity is the sum of all lung capacities, all lung volumes. Okay? It's the sum of all lung volumes. Asbestos fibers penetrate the level of respiratory bronchioles. That is correct. They can go to alveolar level, right? They go to the level of, yes, of course, they go to the level of respiratory bronchioles. Can't they go to alveoli? They can go to alveoli, yes, I know. Asbestosis. Yeah, they penetrate the level of respiratory bronchioles. So that is, respiratory bronchioles is just above the level. So this is the alveolus now. The respiratory bronchiole is the lowest level of the branching conducting system. Okay, so the branching system. This is the alveolus. This is the respiratory bronchiole. We sometimes consider respiratory bronchioles are also involved in a bit of gas exchange, right? They are sometimes exchanged in a bit of gas exchange as well. Yeah, so you can take it, it's not wrong. During homeostatic control of breathing, medulla receives signals only from major blood vessels. Medulla එකට signals එන්නේ කොහෙන් කොහෙන්ද? Where does the medulla receive? It receives so many things. Sensors from the lung tissue, stretch sensors are there. Then medulla itself has sensors. Major blood vessels have sensors, right? So that is false. Only from major blood vessels? No. You get stretch receptor signals from your lungs. You have pH detecting chemoreceptors in the medulla itself. Okay. Any questions? I'll just show you a bit of, hold on. Okay, right. Yeah, no need to complicate you that much at this point. Okay, right. Anyway, right, let's move on. Correct the statement regarding acquired immunity or specific immunity. Haridu? T lymphocytes recognize antigens present in lymph and interstitial fluid. What do you think? T lymphocytes only recognize antigens present in these fluids? No, right? T lymphocytes usually don't recognize things in the fluid. Neither. That is something for the B lymphocytes, right? B lymphocytes bind with antigens presented by macrophages. B lymphocytes are not that picky නේද? Usually our T lymphocytes වලට තමයි මෙහෙම ගිහිල්ලා ඔන්න මෙන්න දිහන antigen කියලා you have to present them the antigen. ඔන්න ගන්න antigen එක කියලා. Okay? Right? So usually T lymphocytes do not I mean B lymphocytes do not need. Okay? Then in the immunity response, okay, in the immunity response, plasma cells in body fluids produce toxins. Plasma cells produce toxins? No, no. Plasma cells produce antibodies that neutralize the toxins. They produce antibodies that neutralize the toxins. Helper T cells may produce secondary immune responses. What do you think? Uh, Secondary immune response means after a subsequent infection of the same antigen, for example, imagine influenza or something, ආපහු හැදුනොත්, chicken pox or whatever, helper T cells can have an influence, but it is mainly by memory, no? It is by mainly by memory T cells. Okay, secondary immune response mainly by memory T cells. Okay, so that is also false. Not helper, memory. Antigen receptors of T and B lymphocytes can be identical, yes. So a single antigen can sensitize both T and B lymphocytes. A single antigen, I mean, a single pathogen can sensitize both T and B lymphocytes to lead into a cell-mediated response as well as a antibody-mediated response for the same pathogen. Okay, simple question. Okay, all right. Reabsorption list. Okay, ටිකක් අමතක වෙන එකක් නේ. So I suggest that you have a diagram of your nephron with the descending and the proximal and the distal and the collecting duct, and have it on your wallpaper, as a wallpaper on your study room, so that you can remember how things are secreted and reabsorbed at different locations. Okay, so that also, if you can see it every day, somehow you are going to remember. That is the strategy. Repetition. Okay, reabsorb and their locations. Active transport of sodium, proximal convoluted tubule, PCM, for PCT. Do you have active transport? Yes, there is a transporter. Okay, selective reabsorption of sodium happens. That is correct. Passive transport of sodium, ascending limb of loop of Henle. What is going to happen in the ascending and descending? In the descending limb, there is water reabsorption. You lose water. In the descending limb, you lose water. So, when it is this urine is traveling up the ascending limb, it is more concentrated. It is more concentrated. So, you lose water in the descending limb, the ascending limb is impermeable to water. What's going to happen? Because it's more concentrated, sodium is going to go out passively. Sodium is going to go out passively in the ascending limb. So, that is also correct. Bicarbonate, passive reabsorption of bicarbonate, that is a theory point that you need to remember, PCT. Passive transport of water, descending limb of loop of Henle, yes, water passively is reabsorbed in the descending limb because the descending limb is water permeable. Passive transport of Cl minus, proximal convoluted tubule, yes, because the sodium is being actively reabsorbed. Sodium ge යාළුවා කවුද? Chlorine. So chlorine is going to come out wherever sodium is going out because it's a positive negative charge relationship. So ඔක්කොම ටික හරිනේ. A, B, C and D. Okay. Clear. Right. So the plan is we'll discuss the rest of the questions next week. Okay. And submit your papers to be marked, upload your papers to be marked. If you want, you can come to ISM and write the papers. Okay, I will give you the printed papers. You can collect from the office and you can write the papers, and they will be also be marked. Okay, so we'll stop for the day here. So keep on studying, right? And from this point onwards, we are going to, the revision class, there's a separate plan for the revision class. I'll share that with the group. So stay updated with the group, with everything that is happening. Okay, so yeah, any question you want to clarify? Any doubts? Any problems? So there's going to be a microbiology special tute that will be delivered to you. Okay. All right, we can finish for the day. Then thank you for joining. Okay, thank you. How to keep everything you memorize. Repetition. Systematically you have to repeat and You have to apply it again and again. You have to apply. I know it can be tiring, but that is the only way. Yes. Okay. You're welcome. Good night.